Hello. Very good evening to the students who have joined. Can you let me know in the chat box if my voice is audible to you? And also, please let me know if the presentation and the video is visible to you. Let me know. Okay, thank you for confirming. Please wait till it's 6 p.m. Then we will start today's session. Till then, please wait. If you have any doubt or any queries from the previous class, you can let me know now. Okay. In the last class, we have discussed about the chemistry of the cofactors and coenzymes. We have discussed a lot about different coenzymes, cofactors, and how they are involved in various biological systems. Today's topics are a little different. We earlier we were discussing about the organic chemistry of the various biological molecules and various biological systems, how the chemistry is related to all these things. We were discussing all those things in our previous 8 classes, but today's topics are a little different. So I hope you will understand these things also. Let us wait for 5 more minutes, then I think I will start the session. Let us wait for 2 more minutes, then I will start the session. Okay. As I have already mentioned, if you have any doubt or any query from the last class, you can let me know now, or you can let me know through email also. Okay, so it's 6 p.m. and I think I will start our session. Let me present the presentation. Okay, so let me make it a big screen. So very good evening to the students who have joined. My name is Mousumi and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAYAM course of Organic Chemistry in Biology and Drug Development, and today it is our week 9 live session. So before starting the session, let me clarify a few things, that there will be a problem solving session every week, and these sessions are scheduled on every Friday from 6 to 8 p.m. The sessions are live on Google Meet platform. This is the Google Meet link which you have used for joining the class today, and this link will remain same for our entire course. Okay, so you can write it down or save it for somewhere, and otherwise you are always reminded by the NPTELs before day before of the class, so that you can use for joining. So the important thing is, if any network disruption happens or any kind of issue happen from my side, please do not leave the session. I will try to rejoin as soon as possible. Okay, I am recording the session. This recording I will be sharing to you after the session. So since I am recording the session, and so that I can record it seamlessly, I have muted your microphones and cameras. But I have here the chat box is open in front of me. So if you have any doubt or any query, you can let me know through the chat box. I am available through the chat box in our entire session. We will use this chat box for using our for question answer purpose and everything. Okay, so you can contact communicate me through the chat box, and during the session you can post your doubts, your query, everything to the chat box. And if still after the class your doubt persists, you can drop an email to me, and I will answer your query through the email also. This presentation and class recording I will be sharing to you after the session. Now, where you can find these things, you can go to the NPTEL SWAYAM portal. Okay, where the course contents are released every week. Then you will find here, you will be finding there is a problem solving session tab. If you click on that particular thing, you will find there is a problem solving session recording. It will be redirecting to you to a page where you will find the lecture videos, and also the it will contain a Google Drive link. The Google Drive will contain all the previous classes, and the ongoing class link also I will be posting after the session. Okay, so these are the things which I had to say before starting the session. So yeah, now let us so today it is our week 9 session. So already we have completed our 8 week course till now, and in this 8 weeks basically what we have done, we have discussed mainly about the biomolecules. Okay, like in our week 1 we have discussed about the structure of amino acids, the zwitter ions, how they behave, the amino acids. Then we have shifted to the proteins, their structure, their functions, followed by enzymes, coenzymes. We have discussed about the central dogma, about the DNA, RNA, the nucleic acid synthesis, how it happens, and how the protein synthesis happens with the help of the mRNA, and all the things that we have already discussed. We have discussed about various things like molecular or aspects of molecular biology, like restriction enzyme, DNA combinatorial, DNA synthesis, recombinant DNA synthesis, and all these things. So till now what we were discussing, all these are related to the various biomolecules or bioenzyme, what it happens in case of our biology. So now onwards what we will be discussing, it is more related to the pharmacological backgrounds, or from or the medicinal background. Okay, so this thing which we have, so upcoming 4 classes we will utilize for discussing about various therapeutics, various therapeutic reagents, and all these things. But today the topics are a little different, difficult. I mean different from what we were discussing from our previous 8 sessions. Okay, so first let me show you what are the topics of today's session, and then let us go to our question answer session. So every class what I do is like I show you around 20 MCQ questions from that particular week's course content. All question contains 4 different options, and you have to identify A, B, C or D, which of the question answer will be correct, that you have to identify, and that particular you have to let me know through in the chat box. Okay, and after you try, then I will show you what is the correct answer and we will discuss that particular topic in detail. Okay, that's how we do in every session. So today it is our week 9 live session. So let us first see what are the topics of the week 9 course content. So the week 9 course contents are like this. We have introduction to the drug discovery process. We have fundamental principles of drug discovery process. We are just going to discuss about combinatorial chemistry, neurotransmitters, catecholamine based and GABA neurotransmitters. We will discuss about hypertension, how the humoral mechanism and renin-angiotensin system mechanism happens, and we will discuss about the inhibitor design of the angiotensin converting enzyme. So these are the topics of the week 9 our course content that we are going to discuss today. So let us start with the first question of today's session. So the first question of today's session is, it is the correct order of the drug discovery program. So your number A, number 1 is the target discovery, number 2 is the lead identification, number 3 is the lead optimization, number 4 is the hit identification, and number 5 is the preclinical development. So these are the 5 steps. Now you have to identify what is the correct order of the drug discovery program. Your first option number A is 1, 4, 2, 3, 5. Then number B is first is 2, then 1, 3, 4, 5. Number C is 3, 4, 1, 2, 5. And number D is 1, 3, 4, 2 and 5. So what will be the correct order of drug discovery system? 1 is given like this, your target discovery. 2 is the lead identification. Number 3 is the lead optimization. Number 4 is the hit identification. And number 5 is the preclinical development. So you have to identify what is the correct order of the drug discovery program. Your given options are: number A is 1 followed by 4 followed by 2 followed by 3 followed by 5. Number B is 2 followed by 1 followed by 3 followed by 4 then 5. Number C is first 3 then 4 then 1 then 2 then 5. And number D is 1 then 3 then 4 then 2 then 5. Let me know A, B, C or D, which of the following you think is the correct answer. I will wait for 2 minutes, then I will check the chat box, read the all the top points carefully, and then let me know what will be the correct answer. Since there are no marking system, so you can answer freely. Even if your answer is wrong, there is no problem. But try to answer the questions. What will be the correct order of the drug discovery program? Okay, let me check the chat box. Let me see if there is any answer given in the chat box for problem number 1. Okay, I got two answer. One is saying it is number D, another is saying it is number B. Now let me check which of the following will be correct, or is there any different answer. So the correct answer for problem number 1, it will be, it is number A. Okay, first is the your target discovery, followed by your hit identification. Then what will happen? Lead identification, then lead optimization, then preclinical studies. Okay, so this is the correct order: 1 followed by 4, followed by your 2, then 3, and then 5. Okay. So first is the target discovery or validation first. So before a drug discovery program, so first what you have to do, you have to identify what is the disease and what is the target. Okay, that is the first obvious step of a drug discovery program. That what the disease, what we are targeting for developing a drug. Let's say if we are targeting malaria. Okay, Then first you have to identify what should be the target, because a particular disease like malaria or like cancer, they can have multiple targets. Okay, always there are multiple targets associated with a particular disease. There is no single target diseases are there. Okay, every disease is associated with a multiple targets. So first is thing is that you have to do is discover the target and validate that is the target is sufficiently important for that particular disease. So that is the first step. Followed by is the hit identification, then lead identification, lead optimization, and preclinical development. So target discovery and validation is the target ID or target prioritization happens, disease target association, and ligandability assessment. That is the first step. Okay, followed by hit identification. So how the hit identifications can be done? By protein modeling, virtual screening, or library enumeration. Okay, then followed by the lead identification. How lead identification can be done? By scaffold hopping, binding mode prediction, physical chemistry property prediction, synthesis planning, in silico admit. All these things I will discuss in the step by step. Okay, so you don't have to worry about these things. Then lead optimization happens. What is this thing? There is a multi-parameter optimization, prediction of the DMPK endpoints, generate modeling, PK/PD, and PBK/PK modeling. So these are the lead optimization. And then we have the preclinical development where the dose prediction happens, then translation and biomarker discovery also is done. So all these are the steps for the target discovery and identification. So first is the target validation. Then you have to identify the hits, and this step involves the computational studies also. Then we have to identify the lead molecules, and then we have to improve the lead molecule by optimizing various properties, and the synthesis also. Then is the preclinical development which happens. Okay, so what are the key points? The key points, like pharmaceutical companies tend to concentrate on developing drugs for disease which are prevalent in the developed countries and aim to produce compounds with better properties than the existing drugs. A molecular target is then chosen which is believed to influence a particular disease when affected by a drug. The greater the selectivity that can be achieved, the less chance of the side effects. A suitable bioassay must be devised which will demonstrate whether a drug has activity against a particular target. Bioassays can be carried out in vitro or in vivo, and usually a combination of the tests is used. HTS involves the manipulation or automation of the in vitro test, such as this is the high throughput screening. Okay, HTS, such that a large number of tests can be carried out in short period of time. Compounds can be tested for their affinity to the macromolecule target by the NMR spectroscopy. The relaxation times of the ligand bound to the macromolecule are shorter when they are unbound. SPR, SPA, ITC are the three visual methods which are for detecting whether the ligands to bind to the macromolecule target. Virtual screening can be also used to identify compounds most likely to be active in the experimental screening. So all these things are related to our drug discovery program. Now one thing is very important, which you have to remember and which you have to understand for a drug discovery program, that is what is a lead compound. Lead compound is a very important term, and you will be hearing this term for a couple of times during our today's session and upcoming sessions also. So what is a lead compound? A lead compound is a structure which shows a useful pharmacological activity and can act as a starting point for the drug design. Natural products are rich source of lead compounds. The agent responsible for the biological activity of a natural extract is known as the active principle. Lead compounds have been isolated from plants, trees, microorganisms, animal venom, toxins. A study of the medical phosphor indicates the plants and herbs which may be contain as novel lead compound. Lead compounds can be found by screening synthetic compounds obtained by combinatorial synthesis and other sources. Existing drugs can be also used as the lead compounds for the design of the novel structures in the same therapeutic area. Alternatively, the side effects of an existing drug can be enhanced to design the novel drugs in a different therapeutic area. Okay, so these are the key points related to our lead compound. Okay, so is it clear or is there any doubt? If there is any doubt, please let me know in the chat box. Okay, so correct answer was number A. First is the target discovery, followed by hit identification, lead lead optimization, and then is the preclinical development. Now let me go to the next problem. Problem number 2, it says that Which country is world's largest producer of generic drugs? Your options are number A USA, number B China, number C India and number D Japan. Which country is world's largest producer of the generic drugs? USA, China, India or Japan? Let me know A, B, C or D which of the following should be the correct answer. I will wait for a minute, then I will check the chat box. Which country is world's largest producer of generic drugs? USA, China, India or Japan? Which country is world's largest producer of generic drugs? USA, China, India or Japan? Let me check the chat box. Let me see if there is any answer. Okay, I got two answers and both are saying that number C is the correct answer. Let me check if it is correct or not. So the correct answer will be yes, it is number C. India is the world's largest producer of the generic drugs. So, what is a generic drug? Let us first understand that. So, generic drugs are the copies of the brand name drugs that have the same active ingredient, strength, safety, and quality and effectiveness. They become available in the market after the original brand name drugs patent expires. Okay. So, what is the as the differences and of the aspects? So generic drug active ingredient must have the identical active ingredient at the brand name version. The brand name drugs contains the original patented active ingredient. Okay, so every drug contains the active pharmaceutical ingredient or API, and the original brands of the particular drug. When first time it comes to the market, it is patented, and only they can use it. But after the patent expires, other companies also can make it under different name but using the same active ingredients, strength, safety, and quality. These drugs are known as generic drugs. Okay, inactive ingredient may differ. Inactive ingredients such as the flavoring, filters, fillers, and colors may have different inactive ingredient than their generic counterparts. Price significantly less expensive because manufacturers don't have the repeat the costly research and development that went into the original drug, and the brand name drugs are more expensive because of the high cost of the initial research development and marketing. Okay, so that so why sometimes you can wonder that why generic drugs are cheaper than the brand name drugs, because the brand name drugs, when a particular drug first comes to the market, it takes a long time, long research and development, clinical trials and everything now with the particular drug. That is why it is more expensive. But in case of the generic drug, since the active ingredient is same, the this all this research development marketing it is not necessary. That is why it has significantly less expensive than the brand name drugs. Appearance may look different in shape, color, size due to the trademark laws and that prevent them from looking exactly same as the brand name drug. And the brand name drugs have the distinctive trademarked appearances. Regulation must meet the same religious quality standards set by the regulatory agencies like US FDA or US Food and Drug Administration must meet the same high standard for safety and effectiveness. Bioequivalence may be bioequivalent, meaning they are absorbed into the bloodstream at the same rate and the same extent to the brand name drug. The brand name drugs are the original product against which the generic versions are measured for the bioequivalence. So brand name drugs are the standard for the calculating the bioequivalence of the generic drugs. Okay. So how generic drugs are approved? To be approved, the generic manufacturers submit an abbreviated new drug application or ANDA to FDA. The abbreviated process allows the manufacturers to avoid the repeating expensive and time-consuming animal and human clinical trial by demonstrating that their product is bioequivalent to the already approved brand name drug. Okay. So ANDA must include the data of the following. The active ingredient is the same. It is identical in strength, dosage form and route of administration. The inactive ingredients are safe. The drug is stable and effective over its shelf life. The manufacturer adheres the same good manufacturing practices required for the brand name drug. So these are how the generic drugs are approved. So the generic drugs or generics is a drug which is produced and distributed without the patent protection. The generic drug may still have the patent on the formulation but not on the active ingredient. A generic must have contain the same active ingredient as the original formulation. When the generic products became available, the market competition often leads to substantially lower prices for both the original brand name product and the generic forms. In the US, FDA US drug patents given for 20 years of protection, but they have applied for before clinical trials begin. So the effective life of the drug patent tends to between 7 to 12 years. And India is world's largest manufacturer for the generic drug. This is very important thing, and it is a very good thing that India is the largest manufacturers of the world's largest manufacturer of the generic medicine. And the companies which are involved in the formulation of this and for manufacturing of the generic medicines are like Sun Pharma, Dr. Reddy's, Cipla, Lupin, Zedios, Aurobindo Pharma, Glenmark, Torrent Pharma, Ranbaxy, and Natco. Okay. So is it clear what is a generic drug and how it is different from the original drugs? Is there any doubt? Okay. Then I will move toward our next problem. Now let me move to the next problem. Problem number 3. It says that when a drug is approved, it says that when a drug is approved for the human testing during clinical trials, your options are number A between phase 3 and phase 4, number B between phase 1 and phase 2, number C at the end of the phase 4, and number D at the end of the preclinical trial. So when a drug is approved for the human testing. Your options are number A between phase 3 and phase 4, number B between phase 1 and 2, number C at the end of the phase 4, and number D at the end of the preclinical trials. I will give you a minute, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 3. When a drug is approved for human testing during the clinical trials, option number A between phase 3 and phase 4, option number B between phase 1 and phase 2, option number C at the end of the phase 4, and option number D at the end of the preclinical trial. When a drug is approved for human testing during clinical trial, let me check the chat box now. Okay, I got two answers. One is saying it is number C, other is saying it is number A. Let me check which of the following is the correct answer. So, a drug is approved for the human testing during So correct answer is number D, at the end of the preclinical trial. Okay. So I think like in the last slide, which I have mentioned that the correct order of drug discovery is like this. So at the end, we have this preclinical development, and after this preclinical development, the human testing begins, which is known as the clinical trials. Okay. So what is the clinical trial process? The first is the preclinical trial, exploring if the new drug how the new drug may work. Okay. During the and after that, the phase 1 clinical trial started. After a drug passes the preclinical studies, it is approved for the human testing, and human testing is it happens in fourth step. In the first phase 1, exploring the safety and dosing of the drug. Okay, and phase 2, exploring the effectiveness of the drug. The phase 3, exploring the safety effectiveness compared to the currently available treatments. Okay, and all this clinical trial, it is done on the volunteers, the human volunteers who have given their consent for the clinical trials of particular drug, and it may take up to 5 to 15 years. Okay, and special designations can speed up the process of the clinical trial, like we have during our COVID-19 pandemic. Okay, because in very short time we have to approve the vaccines, because it was a special situation. After the phase 3, the particular after a drug this passes the all the phases of the drug discovery, up to this, it is approved from the FDA approval. FDA is the Food and Drug Administration, which is the governing body of US. Every drug have to pass the FDA approval before coming into the market. To confirm the safety and effectiveness of the drug, the FDA review happens, then the drug is approved. After some time, when the drug is in market and the drug is used in a larger population of people, a phase 4 clinical trial is also happen, which evaluates the drug over time and large number of patients. Okay, sometime it may happen the clinical trial, although it takes up very much a lot of time and many human volunteers, but when a drug comes to the larger population, it may tend to develop some kind of side effects, or over the period of time its effectiveness might get reduced. All these things are discovered during our phase 4 clinical trial. Okay, so these are the processes, different processes of clinical trial. So what happens in the preclinical trial, that you have to know first. Okay, so the preclinical trials or non-clinical trials are the laboratory test for a new drug, substance, or medical device, usually done on an animal subject, to see whether the treatment is really works or it is safe to test on the humans. The main goals of the preclinical studies are to determine the product's ultimate safety profile. The products may include new medical devices, drugs, gene therapy solution, etc. Objectives of the preclinical trial: the purpose of the preclinical trial is to develop adequate data to decide that it is reasonably safe to proceed with the human trials of the drug. Means laboratory test of a new drug or a new medical device, usually done on the animal subjects, to see if the treatment really works or if it is safe to test on the humans. However, the main objective is to collect the data to submit from the FDA for IND filing. And importance of the preclinical trial: to determine the dose, toxic dose, pharmacological action, etc. It is the requirement of the regulatory body for performing the clinical trials. As an ethical ethical viewpoint, it is necessary to safety check the drugs on animals before starting on the human being. To check the kinetic profile of the drug, based on it, select the route of administration in human for the clinical trials. Okay, so remember, a drug is approved for the human testing at the end of the preclinical trial. Okay, and the drug is approved for the FDA after the submitted for the FDA approval at the end of the phase 3, and after the drug is approved, the phase 4 clinical trial, it is done. Okay, so is it clear, or is there any doubt? If there is no doubt, then I am going to proceed to the next question. Okay, whatever doubts you have, you let me know through the chat box. I will discuss it in meantime, or we can discuss it at the end of the session also. Okay. So let us go to the next problem. So problem number 4, it says, which of the following terms is for the therapeutic index of a drug for humans? Your options are: number A, TD50 by ED50, number B, LD50 by ED99, number C, IC50 by TD50, and number D, EC99 by IC50. What is which of the following terms is for the therapeutic index of the drug for humans? What is the which denotes the therapeutic index of the drug for humans? Number A, TD50 by ED50. Number B, LD50 by ED99. Number C, IC50 by TD50. And number D, EC99 by IC50. Let me know A, B, C or D, which of the following is correct for problem number 4. I will wait for a minute, then I will check. Let me know which is the correct indicator of the therapeutic index of a drug. Let me check the chat box now. Okay, I got answer and both are saying that it is number C. Let me check if number C is the correct answer or not. So the correct answer will be, it is number A. TD50 by ED50 is the therapeutic index of the drugs in humans. Okay. So this is the drug safety profile. First, let me, yeah. So this is this y-axis indicates the percentage of responding from 0 to 100, and this is the concentration of the drug mg to kg. So this is the therapeutic effect, and this is the toxic effect. Okay. So what is the terms? What is the TD50? TD50 is the toxic dose in the 50% of population. Okay. So the concentration of drug that is toxic for the 50% of population is known as the TD50, and ED50 is the concentration of the drug which is required to be effective in a 50% of population. And IC50 actually TD50 and LD50 is the same thing. TD50 is the toxic dose, LD50 is the lethal dose. Lethal dose, it is basically LD50 term, it is used for animals, okay, whereas the TD50 is used for human. IC50 is another term. IC50 is the inhibitory concentration inhibitory concentration of the 50%. So to make a particular drug, enzyme to inhibit its activity to 50%, the concentration is known as the IC50 value. So the therapeutic index of the is the quantitative measure of the drug safety, calculated as the ratio of the drug that produces a toxic effect to the dose that produces the desired therapeutic effect. Okay, a higher therapeutic index indicates a safer drug with a wider margin between its effect and the harmful doses. So the standard formula of therapeutic index, or TI, is the median toxic dose and the median effective dose. Is the TI therapeutic index is the TD50 by ED50, where ED50 is the dose at which 50% of the population achieved the desired therapeutic effect. TD50 is the dose at which 50% of the population experiences a toxic effect. The preclinical animal studies, the lethal dose is sometimes used in the case of the TD50. Okay, and this curve also, it shows that this is a for the toxic side effect, this drug, and this is for the therapeutic effect. So this is the ED50, now where the concentration of drug that is required for the 50% of responding. This is the TD50, which is the 50% of the concentration of drug that is required for the 50% of toxicity. Okay, and so therapeutic index is TD50 by ED50. So what does this high therapeutic index mean? High therapeutic index indicates a wide safety margin. A drug with a high therapeutic index considered to be safer because the wide range dosage between the effective and toxic dose. For example, let's say the concentration of a drug is 2 mg per kg for the effective, and for the toxic it is 200 mg per kg. So if it is then what so what is the therapeutic index? It will be 200 divided by 2 is the 100. But if that I mean ED50 is let's say 2, and if TD50 or toxic dose is let's say 100, so you see what the I mean the safety margin is decreasing if the therapeutic index is low. Okay, so high therapeutic index indicates that the drug is safe because the wide doses range between the effective and toxic dose. Healthcare providers can prescribe these drugs with less concern for the close monitoring, as there are low risk of the toxicity and effective doses. For example, penicillin has a high therapeutic index. Low therapeutic index or narrow safety margin. A drug with a low therapeutic index is a has a narrow safety margin, meaning effective dose is close to the toxic dose. Even small increase of the dosage can lead to the toxic effects. Patients on this medication use careful dosing and frequent monitoring, often through the blood test, to ensure drug stays with a safe range. Example, rigoxin, warfarin, lithium are well-known narrow therapeutic index drugs. Warfarin is the vitamin K antagonist, which we have discussed in our last class, if you remember. Important considerations is the therapeutic window. A related term, the therapeutic window refers to the range of the drug concentration in the blood that provide a safe and effective treatment. Individual variability. A drug's therapeutic index is an average measurement for a population. Individual patient factors such as age, genetics, and other medical conditions can alter the drug's effects. Complex interactions. A drug-drug interactions or the presence of the substances like alcohol can change the drug safety profile and affects its therapeutic index. Okay. So these are the important considerations. So is it clear what is therapeutic index, or should I repeat? So therapeutic index is the ratio between, so remember it is the ratio between the median toxic dose and the median effective dose. Okay. Toxic dose, median toxic dose means the dose at which 50% of population experiences toxic effect. Median effective dose means the dose at which 50% of the population achieve the desired therapeutic effect. And high therapeutic index indicates that the particular drug has a wide safety margin, whereas the low safety margin indicated by the low therapeutic index. And in this low therapeutic index cases, the dosage should be monitored very carefully, because a little increase of the dosage can lead to the side effects. So I hope it is clear what is the therapeutic index and how it is measured. There can be mathematical questions can also be given. For example, you can be given the ED50 and TD50 values, and you may be asked to calculate the therapeutic index. So I think you can do it now. Let me go to the next problem. Problem number 5, it says, which organ is known to be body's customs office? Your options are number A, heart, number B, brain, number C, pancreas, and number D, liver. Which organ is known to be body's custom office? Heart, brain, pancreas, or liver? Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 5. I will wait for a minute, then I will check the chat box. Meanwhile, I want all of you to answer. Let me know which of the following will be the correct answer for problem number 5. Which organ is known to be body's custom office? Heart, brain, pancreas, or liver? Let me check the chat box. Let me check if I have any answer. Okay, I got two answer. One is saying it is number B, another is saying it is number D. Let me check which of the following is the correct answer for our problem number 5. So the organ which is known as to be the body's customs office will be, yes, the correct answer is number D, it is liver. Okay. Now, why liver is called body's customs office? So, let us first understand what is the typical journey of an orally active drug. So, the preferred method of the drug administration is the oral route. So, we shall consider some of the hurdles and the hazards faced by the certain drug in order to reach its eventual target. When a drug is swallowed, it enters the GIT, which is the gastrointestinal tract, which comprises with the mouth, throat, stomach, and the upper and lower intestine. A certain amount of drug is may be absorbed through the muscular membrane of the mouth, but most of them passes down to the stomach where it encounters gastric juices and the hydrochloric acid. These chemicals aid the digestion of the food and will treat a drug in the similar fashion if it is skeptical to break down, but it is not protected within the acid-resistant pill or the capsule. For example, first clinically useful penicillin was broken down in the stomach and had to be administered by injection. Other acid-labile drugs include the local anesthetics and insulin. If the drug does survive the stomach, it enters to the upper intestine where it encounters the digestive enzymes that serve to break down the food. Assuming the drug survives this attack, then it has to pass through the cell lining of the gut wall, which means that now it has to pass through the cell membrane of the two occasions, first to enter the cell and then to exit the other cell. Now, once the drug has passed through the cells of the gut wall, it can enter to the blood supply relatively easily as the cells lining of the blood vessel are loose fitting and the pores through which most of the drugs can pass. In other words, the drug enters the blood vessel by passing through the cells rather than through them. Then the drug is now transported to the blood to the body's customs office, the liver. The liver contains enzymes that are ready to waiting to intercept the foreign chemicals that modified them such that they are more easily excreted, a process known as the drug metabolism. Following this, the drug now has to be carried by the blood supply around the body to reach its eventual target, which may require crossing further cell membranes, always assuming that it is neither excreted before it gets there nor it diverted to the other parts of the body where it is not needed. It can be seen that the stringent demands are made on the any orally administered drug. It must be stable to both chemical and enzymatic attack. It must also have the correct physiochemical properties to allow it reach its target of the therapy in the therapeutic concentration. This includes the efficient absorption, effective distribution of the target tissue, and is an acceptable rate of the excretion. So these are the demands or these are the important things which are required by a orally active drugs. Okay, so this part is very necessary to understand and to remember what are the requirements of a orally active drugs to reach to its therapeutic site. Okay, so yes, the correct answer was number D. Liver is the body's customs office. Now I will go to the next problem. Now, problem number 6, it says that which of the following statements is true regarding Lipinski's rule? Your options are number A, there is no exceptions to this rule. Number B, hydrogen bond donor and acceptor value must be equal to 5. Number C, cyclosporine does not obey this rule. And number D, all of the above. Which of the following statements is true regarding Lipinski's rule? Again, I will repeat number A, there are no exception to this rule. Number B, hydrogen bond donor and acceptor value must be equal to 5. Number C, cyclosport does not obey this rule. And number D, all of the above. Which of the following statements is true regarding the Lipinski's rule? Let me know A, B, C or D, which of the following will be the correct answer. Which of the following statements is true regarding Lipinski's rule? Read the options very carefully because this kind of true false question sometimes it may be tricky. So read the options carefully and then let me know A, B, C or D which of the following will be the statement that is true regarding Lipinski's rule. Let me check the chat box. Let me see. Okay. I think I got two answers and both are saying that number B is the correct answer. Let me check if number B is the correct answer or not. So the statement which is true regarding the Lipinski's rule will be, it is number C. Cyclosporine does not obey this rule. First, let us understand what is Lipinski's rule. So, Lipinski's rule of five. These are the which, so for a drug to be, to be orally active. So these are should be the considerations. First is not more than five hydrogen bond donors. Okay. So hydrogen bond donor number should not exceed the value of five. Not more than 10 hydrogen bond acceptor. Okay, so hydrogen bond acceptor values may be more than 5, but it should not be more than 10. Okay, the molecular weight is less than 500 and C log P value not greater than 5. So these are the Lipinski's rule. Which are the these are the Lipinski's rule. So now let us go to the statement. So for the first statement, it says that there are no exceptions to this rule. This is incorrect. There are several exceptions of the Lipinski's rule of 5. Number B, hydrogen bond donor acceptance value must be equal to 5. It is also incorrect because here I have already shown you that the hydrogen bond donor not more than 5 hydrogen bond donor. Okay, so hydrogen bond donor value may be less equal to 5. Okay, and hydrogen bond acceptor value should be less equal to 10. That is why statement number B is false. Molecular weight should be less than 500 and C log P value, it should not greater than 5. So these are the considerations of Lipinski's rule. Now statement number C, which says cyclosporine does not obey this rule, this is correct. Why this is correct? Now let us discuss it. So the hydrophilic and hydrophobic character of the drug is crucial factor affecting the absorption through the gut wall. In theory, molecular weight of the drug should be irrelevant. For example, cyclosporine is successfully absorbed through the cell membrane, although it has a molecular weight about 1200. This is the structure of the cyclosporine. It has a molecular weight about 1200. Still, it can easily successfully absorb through the cell membranes. Okay. In practice, however, larger molecules tend to be poorly absorbed. As a rule of thumb, orally absorbed drugs tend to obey. That is known as the Lipinski's rule of 5. The rule of 5 was derived from the analysis of the compounds from the World Drug Index database and aimed at identifying features that were important for making a drug orally active. It was found that the factors concerned involving the number are multiples of 5. Okay, like a molecular weight less than 500, no more than 5 hydrogen bond donor group, not more than 10 hydrogen bond acceptor groups, a calculated log P value less than plus 5. Okay. This log P is a measure of the drug's hydrophobicity. The rule of 5 has been an extremely useful rule of thumb for many years. It is neither, okay, 1 second. Yeah. So this rule of 5 has been an extremely important thumb rule for for many years, but it is neither quantitative nor foolproof. For example, orally active drugs such as atorvastatin, rosuvastatin, cyclosporine, vinorabin do not obey this rule of 5. It has also been demonstrated that a higher molecular weight does not itself cause poor oral bioavailability. One of the reasons that the molecular weight appears to be important is the large molecules are invariably have too many functional groups capable of forming hydrogen bonds. Another source of the debate concerned the calculation of the number of the hydrogen bond acceptors. In Lipinski's original paper, number of hydrogen bond acceptor corresponded to the total number of oxygen and nitrogen atom present in the structure. It was done for the simplicity's sake, but most medicinal chemists would discount weak hydrogen bond acceptors such as the amide nitrogens. Therefore, it is better to view Lipinski's rule as the set of guidelines rather than the rules. Lipinski himself stated that the compound was likely to be orally active as long as it does not break more than one of his rules. Okay. So statement number C was the correct statement for problem number 6. Now I will go to the next problem. Problem number 7, it says identify the correct statement about the combinatorial chemistry. Your options are number A, one molecule can be done at one time. Number B, it has a high risk of failure. Number C, it was developed using the solution phase synthesis. And number D, it was developed using solid phase synthesis. Which of the following statement is correct regarding the combinatorial chemistry? Read all the options very carefully. Then let me know A, B, C or D, which of the following you think is the correct statement for combinatorial chemistry. Again, I will repeat. Number A, one molecule can be done at one time. Number B, it has a high risk of failure. Number C, it was developed using the solution phase synthesis. And number D, it was developed using the solid phase synthesis. Identify correct statement regarding combinatorial chemistry. Again, I will wait for a minute or two, then I will check the chat box. Read options carefully, then let me know. Let me check if there is any answer for problem number 7. Okay, I got two answers. Both are saying number D is the correct answer. Let me check if number D is correct or not. Okay, regarding the combinatorial chemistry. So the statement which is correct will be very good. It is number D. It was developed using the solid phase synthesis. Other statements are not correct. Okay. So combinatorial synthesis, so two things are very important in case of the drug discovery and things. So one is combinatorial synthesis and another is the parallel synthesis of the medicine in the medicinal chemistry projects. Okay, so combinatorial and parallel synthesis have become established tools in the drug discovery and development, allowing the use of the defined reaction route to produce a large number of compound in a short period of time. In the full set of compounds produced in this way is called the compound library. Reactions are usually carried out in a small scale, then the process can be automated to a semi-automated or semi-automated, allowing reactions to be carried out in a several reaction vessels at the same time under identical condition, but using the different reagents for each vessel. The compact nature of the apparatus means that the process can be carried out within the normal fume cupboard. Combinatorial parallel synthesis in the past medicinal chemistry involved the identification of the lead compound having useful activity, which was then modified to develop clinically useful drug. Identification of the molecular target for the drug and the mechanism by which it worked often took many years to establish. Today, most of the medicinal chemistry projects start to with an identifiable target, and then the emphasis is on the discovering the lead compound that will interact with this target. Okay, so that we have already discussed that what is the stages of the drug discovery and development as the finding a target, finding a lead, isolating the active structure, identifying the structure, then doing the SAR or structure activity relationship studies on the identified lead structure, and then optimization of the lead. Okay. Then this reversal of the priorities came out as a result of the Human Genome Project and the proteomic revolution that followed. Once the genome was mapped, then a vast number of previously unknown proteins were identified, and all this could be considered to as a potential drug targets. Pharmaceutical companies chemists were faced with the problem of identifying the function of the each target and finding the lead compound to interact with it. Before the advent of the combinatorial chemistry and parallel synthesis, the need of the finding lead compound has become limiting factor in the whole process. Now, with the aid of these techniques, research groups can rapidly synthesize and screen thousands of structures and the new lead compounds, identifying the structure-activity relationship studies and analogs of the grouped activity and minimal side effect. The procedures used in the combinatorial synthesis are designed to produce mixture of different compounds which each reaction vessel, whereas those used in parallel synthesis produce single product and the each vessel. Now, in general, parallel synthesis is favored because it is easier to identify the structures that are synthesized. However, there is still scope in the combinatorial chemistry in finding lead compounds, especially as the procedure can guarantee significantly more structures set in the period of time, thus increasing the chances of finding lead compound. Both methods are generally involve the use of the solid phase technique, which are discussed in the later. Okay. So the statements. Now let us look at the statements. First, one molecule can be done at one time in combinatorial. It is the incorrect. Several molecules can be done at one time. It has a high risk of failure. No, it is also incorrect. It was developed using solid phase. It is also incorrect. The combinatorial chemistry is developed using the solid phase synthesis. We will discuss it in our upcoming slides how it is done. Okay, in detail. So yeah, number D was the correct answer. Now let me go to the next problem. Problem number 8 says, which of the following statements regarding the solid state synthesis is false? Your options are number A cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the solid support. Number C, the anchor should contain reactive functional group. Number D, protective group of functional group should involve in the synthetic routes. Which of the following statements regarding the solid state synthesis is false? Whenever there is a true false kind of question, I have already told you that read the options very carefully and then let me know A, B, C or D which of the following is the correct answer for the problem number 8, means which of the following statement is false regarding the solid state synthesis. Your options are number A, cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the support. Number C, the anchor should contain reactive functional group. Number D, the protective group of the functional group should involve in the synthetic routes. I will wait for a minute and then I will check the chat box. Let me know A, B, C or D which of the following is the correct answer. Which of the following statements regarding the solid state synthesis is false? Your options are number A, cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the support. Number C, anchor should contain reactive functional groups. And number D, the protective group of functional groups should involve in synthetic routes. Which of the following statements regarding the solid phase synthesis is false? Let me check the chat box now. Let me check if there is any answer. Okay, I got one answer which is saying it is number D. Let me check if number D is the correct answer for our problem number 8. So the correct answer, the statement which is false, it will be very good. It is number D. The protective group of the functional groups should involve in the synthetic routes. So the solid state technique, the solid phase techniques can be used to carry out reactions where the starting material is linked to the solid support, such as a resin bed. Several reactions can be then carried out in the sequence of the attached molecule. The final structure is then detached from the solid support. There are several advantages to this. Okay. Since the starting material, intermediate, and the final products are bound to the solid support, excess reagent or unbound byproducts from each reaction can be easily removed by washing the resin. Large or excess of the reagents can be used to drive the reaction to completion or greater than 99% because of the ease with which the excess reagent can be removed. Intermediates can in the reaction sequence are bound to the bead and do not need to be purified. The polymeric support can be regenerated or reused in appropriate cleavage conditions, and suitable anchor or linker groups are chosen. Automation is possible if the combinatorial synthesis is being carried out. A range of the different starting materials can be bound to the separate beads. The beads can be mixed together such that all the starting materials are treated with the anchor reagent in a single experiment. The starting materials and the products are still physically distinct as they are bound to the separate beads. In most of the cases, mixing all the starting materials together in a solution chemistry is a recipe of disaster. With polymerizations and side reactions producing the netarimus, the individual beads can be separated at the end of the experiment to give the individual products. The essential requirement for the solid phase synthesis are a cross-linked insoluble polymeric support which is inert to the synthetic condition, an anchor or linker covalently linked to the resin. The anchor is a reactive functional group that can be used to attach substrate, a bond linking substrate or linker which will be stable in the reaction conditions of the synthesis, a means of cleaving of the product and intermediates from the linker, protecting groups that should not involved in the synthetic route. That is why our statement number D is false, because it said that the protective group should involve in the synthetic routes, but no, the protected groups should not. The protected groups for the functional groups should not involve in the synthetic route. Okay. These are the important considerations or essential requirement for a solid phase synthesis. So what is the solid support? The first example of the solid phase synthesis was the medical resin in the peptide synthesis. The resin involved in the consisted in the polystyrene beads that was styrene is partially cross-linked with 1% of divinyl benzene. The beads are derivatized with a chloromethyl group, that is anchor linker, to which the amino acids can be coupled via an ester group. The ester group is stable in the reaction condition used in the peptide synthesis, but can be cleaved at the end of the synthesis using vigorous acidic conditions like with the hydrofluoric acid. Okay, so the Merrifield resin, how it is used in solid phase peptide synthesis, we already have discussed in our first or second class. I think it is in the second class we have discussed. Okay, so this is the example how it happens. So this is the example of the Merrifield using the solid phase peptide synthesis. Okay, this is the resin bit, this is the Merrifield resin, and that's how the first amino acids are attached, and then deprotection, the end product amino acids is attached with this resin, then deprotection happens with TFA and followed by another amino acid which is end protected, then followed by again another deprotection, and then it gives us this polypeptide chain after the cleavage. Okay, so this is how the solid phase peptide synthesis happens using the Merrifield resin. Okay. So is it clear or is there any doubt? Is it clear about the solid phase synthesis? If there is any doubt, please let me know. Okay, I will discuss it again. Otherwise, let me go to the next problem if there is no doubt. Let me now go to the problem number 9 then, if all is clear in this problem number 8. So problem number 9, it says that which of the following reactions is used in the dynamic combinatorial synthesis of the vancomycin dimers? Your options are number A, Suzuki coupling, number B, Bergman cyclization, number C, olefin metathesis, and number D, Michael reaction. Which of the following reactions is used in the dynamic combinatorial synthesis of vancomycin dimers? Your options are number A, Suzuki coupling, number B, Bergman cyclization, number C, olefin metathesis, and number D, Michael reaction. I will give you a minute or two, then I will check the chat box. Read all the options carefully and then let me know A, B, C or D which of the following will be correct answer. Which of the following reactions is used in the dynamic combinatorial synthesis of vancomycin dimers? Let me check the chat box. Okay, I got two answers. One is saying it is number B, other is saying it is number C. Let me check which of the following is the correct answer. Number B or number C, or is there any other different answer? So the correct answer will be, it is number C, olefin metastasis. Okay. So this reaction is involved in the dynamic combinatorial synthesis of the vancomycin dimers. Okay. So vancomycin is an antibiotic that works because it marks the building blocks required for the bacterial wall synthesis. Cell wall synthesis binding takes place specifically between the antibiotic and the peptide synthesis sequence of the L-lysine D-ALA, which is present in the building block. It is also known to be binding promoters of the dimerization of the vancomycin target complex, which suggests that covalently linked vancomycin dimers might be more effective antibacterial agent than vancomycin itself. A dynamic combinatorial synthesis was carried out synthesizing variety of different vancomycin dimers covalently linked by the bridges with a different lengths. The vancomycin monomers used had been modified such that they contain long chain alkyl substituent with the double bonds at the end. Reaction between two double bonds in the presence of a catalyst that lead to a bridge formation known as the olefin metastasis. Okay, so this is the olefin metastasis where the two double bond at the end terminal double bonds, and they are correct. They are connected together and cis trans isomers are formed. The tripeptide target was then present in the accelerate the rate of the bridge formation to promote the formation of the vancomycin dimer having the ideal bridge length. So it is showing in here. So there is the tripeptide. This is the vancomycin monomer, and then first the ligand is binded with the monomer, then dimerization happen. Okay, and the covalently linked dimers are formed. Now, once they form, the dimers having correct length and substituent are likely to react together to form the covalent bridge. Having established the optimum length of the bridge, another experiment was carried out on 8 vancomycin monomer, which led to the correct end length to tether, but varied slightly to their structure. Now, the mixture of 36 possible products was analyzed by mass spectrometry to indicate relative proportion of each dimer. 11 of 36 compounds were then synthesized separately, and it was found that the antibacterial activity matched with the level of amplification that the compounds present in the greater quantities had the greater activity. So that's how it happens. That first the vancomycin monomers are binding with the tripeptide. After the ligand binding, so this is the vancomycin monomer. Then dimerization happen, and the reaction which is involved here is the olefin metastasis. Okay, vancomycin is a very potential antibiotic, and all these things we will discuss in our next class. I think we have our antibacterial agents. Then we will discuss about this vancomycin and other antibacterial agents and their mechanism of actions in more detail. Okay. So now I will go to the next problem. Problem number 10, it says which of the following statements is not true for an in vivo test? Your options are as follows. Number A, it is more expensive and time consuming. Number B, it is less precise than in vitro test. Number C, it is performed under physiological condition. And number D, a whole living organism is required for this. Which of the following statement is not true for the in vivo test? Number A, it is more expensive and time consuming. Number B, it is less precise than in vitro test. Number C, it is performed under the physiological condition. And number D, a whole living organism is required for the in vivo test. Read all the options carefully and let me know which of the following statement is not true. Okay, please highlight this thing that you have to identify the statement which is not true, means the statement which is incorrect regarding the in vivo testing. Let me wait for a minute, then I will check the chat box. Read the options carefully and then let me know A, B, C or D which of the following should be the correct answer for problem number 10. I will wait for a minute, then I will check the chat box. Okay, read the options carefully and then let me know. Which of the following statement is not true for in vivo test? Let me check the chat box now. Okay. Okay, I got two answer. Both are saying that number B is the correct answer. Now let me check if number B is the correct answer for the problem number 10 or not. So the correct answer means the statement which is not true regarding the an in vivo test will be yes, it is number B. Very good. It is less precise than in vitro test. It is not true. So, a in vivo test is more expensive, time consuming. It is more precise than in vitro test. It is performed under physiological condition and a whole living organism is required for this. Okay. So now we have to understand the different terms. So of what are the regarding the testings. First is the in silico. In silico is performed in a virtual setting, a computer or virtual simulation. Then in vitro means in glass, meaning the study takes place in test tube or petri dish and everything. And in vivo means in life, means the study takes place in the living organism. In vitro versus in vivo. In vitro refers to a phenomenon in which a given procedure is performed in a controlled environment outside the living organism. In vivo refers to a phenomenon in which experiments are performed using a whole living organism. Dead organism and isolated cellular components are used in case of the in vitro. A whole living organism is used in case of the in vivo. Performed under the controlled laboratory condition, the in vitro one. In vivo performed under the physiological condition. In vitro is less expensive, less time consuming, less precise. In vivo is more expensive, more time consuming, more precise. For example, cell culture experiment in petri dish and experiments in test tubes are considered as the in vitro test. Drug testing experiment performed by the using Morgan organisms such as mice, rabbit, and apes are considered as the in vivo testing. In vitro fertilization refers to the artificial fertilization method in which fusion of a male and female gametes occur outside the human body. Regular fertilization mechanism in which the fusion of a male and female gametes occurs within the body is referred to the in vivo fertilization. So in vivo testing systems, in vitro is in glass, in ex vivo is the actually there is another testing which known as the ex vivo when the tissues taken from a living organism, there it is tested. Okay, and then in vivo is the first the in vitro tests are happen, then followed by the ex vivo and then in vivo. In vitro, ex vivo, in vivo, all testings are falls under the preclinical testing, and then a drug which passes through all these phases goes to the clinical trial on the human testing. So in vivo testing plays a vital role in drug discovery involving animal testing and clinical trials. Before a drug candidate is tested in human subject, it must go to the animal testing for further evaluate its toxicity, metabolism, and over efficacy. Vertebrate animal models such as the primates, dogs, rabbits are commonly used as the preclinical in the preclinical studies. Okay. So this was all about the in vivo testing. Now I will go to the next problem. So problem number 11, it says which of the following statements regarding neurotransmitters is true? Your options are number A, they are secreted by the endocrine glands. Number B, glutamate is a type of the neurotransmitter. Number C, they traverse through the blood to act on a distant target cells. And number D, their effects are longer lasting. Which of the following statements regarding neurotransmitters is true? Number A, they are secreted by the endocrine glands. Number B, glutamate is a type of neurotransmitter. Number C, they traverse through the blood to act on a distant target cell. And number D, the effects are longer lasting. Let me know A, B, C or D, which of the following statement regarding neurotransmitter is true. Which of the following statements regarding neurotransmitter is true? Let me check the chat box now. I got one answer which is saying it is number A. Let me check if number A is the correct answer or not. So the statement which is true regarding neurotransmitter will be, it is number B. Glutamate is a type of the neurotransmitter. Okay. So neurotransmitters are the chemical substance released by the electrical impulses into the synaptic cleft from the synaptic vesicle to of the presynaptic membrane. Then it diffuses to the postsynaptic membrane, binds to or activates the receptor present, leading to the initiation of the new chemical signals or the inhibition of the postsynaptic neuron. Examples of neurotransmitters. Sorry. So the examples of neurotransmitters are like adrenaline, which is known as a fight or flight. And they produced in a stressful situation, increases the heart rate and blood flow, leading to the physical boost and the heightened awareness. GABA, which is the gamma aminobutyric acid, which has a calming effect, comes the firing nerves in the central nervous system. High levels improve focus, low level cause anxiety. Also contributes to motor control and vision. Noradrenaline, which is which is important for concentration. It affects the attention and responding actions in the brain, contacts the blood vessel, increasing the blood flow. Acetylcholine, it is important for learning, involved in thought, learning or memory, activates the muscle action of the body, also associated with the attention and awakening. Dopamine is for pleasure, feelings of the pleasure, addiction, movement or the motivation. People repeat behaviors that lead to the dopamine release. Glutamate is for memory. Most common neurotransmitter is glutamate. It involved in learning or memory, regulates development and creation of the nerve impact. We have serotonin for mood, contributes to well-being and happiness, helps in the sleep cycle, digestive regulation, affected by the exercise and light exposure. Endorphins for euphoria, released during exercise, excitement and sex, producing well-being and euphoria and reducing pain. So these are the important neurotransmitters. Okay. Now, what is the differences between the hormone and neurotransmitter? So, hormones are released by the endocrine glands, whereas neurotransmitters are released by the nerve cell. That is why statement number one is not correct, because it says that neurotransmitters are secreted by endocrine glands, but it is not. Neurotransmitters are secreted or released by the nerve cells. Hormones are released by the endocrine glands. Hormones travel through the blood to the act on the distinct target cells. Neurotransmitters travel across the synapse to act on the nearby cells. The effects of the hormones can be slower to build but longer lasting. The effects of the neurotransmitters can be fast to act but shorter lasting. Hormones are a part of the endocrine system. Neurotransmitters are part of the nervous system. So this is how neurotransmitter act as a chemical messengers binding to receptor and trigger this reaction in the cell. So basically this is a neuron, and when the signal is happened, the messengers are neurotransmitters are released, and there is a receptor on every cell, and on the cell, when the receptor is on the receptor, the messenger or neurotransmitter binds, and it triggers the responses inside the cells. Okay, so that's how a neurotransmitter act as a chemical messenger binding to the receptor and triggering different reactions in the cell. Okay, so this was the problem number 11, which has the correct answer number B. Now I will go to the next problem. The problem number 12 says, which of the following is false regarding the renin-angiotensin system? Your options are: number A, aldosterone is a steroidal hormone; number B, vasoconstriction is caused by the action of the angiotensin 1; number C, aldosterone enhances the retention of sodium and water in the blood; and number D, bradykinin is destroyed by ACE or angiotensin converting enzyme. Which of the following statement is false regarding the renin-angiotensin system? Again, I will repeat the options. Number A, aldosterone is a steroidal hormone. Number B, vasoconstriction is caused by the action of angiotensin 1. Number C, aldosterone enhances the retention of the sodium and water in the blood. And number D, bradykinin is destroyed by ACE or angiotensin converting enzyme. Let me know A, B, C or D. Which of the following statement is false regarding renin-angiotensin system? Which of the following statement is false regarding the renin-angiotensin system? Your options are number A, aldosterone is a steroidal hormone. Number B, vasoconstriction is caused by the action of angiotensin 1. Number C, aldosterone enhances the retention of sodium and water in the blood. And number D, bradykinin is destroyed by ACE or angiotensin converting enzyme. Let me check the chat box now. Let's see if there is any answer for problem number 12. Okay, I got only one answer for problem number 12. Okay, let me discuss it. Okay. So the correct answer will be yes, it is number B. Vasoconstriction is caused by the action of angiotensin 1. This statement is false. So this is the renin-angiotensin pathway. First is dehydration happens because or this dehydration may happen, then sodium deficiency or hemorrhage, then which leads to the decrease in the blood volume and decrease in the blood pressure. Once the blood pressure is decreased, the juxtaglomerular cells of the kidneys are activated, which increase the secretion of the renin. And then we have the from liver we have the angiotensin, and this renin and angiotensin increases the increased renin increases the angiotensin 1, and with the help of the angiotensinogen. Okay, this angiotensin 1 is converted to the angiotensin 2 in the lungs with the help of the AC or angiotensin converting enzyme, and this angiotensin 2 leads to the vasoconstriction of the arterioles. Okay, and then it can also lead to the adrenal, it activate the adrenal cortex, and which increases the aldosterone. Okay, and increased aldosterone can lead to in the kidneys for the increased sodium and water reabsorption, which increase the blood volume, and then which increases the blood pressure until it comes to the normal. The vasoconstriction of the arterioles also leads to the increase of the blood pressure. Okay, and increase of the potassium in the extracellular fluid can be taken up by the adrenal cortex, which again increases the aldosterone. So this is the renin-angiotensin aldosterone pathway, which is deeply connected with our blood pressure of our body. And aldosterone is very important because it gives rise to the sodium retention, which leads to the high blood pressure for the oxidative stress, damage of DNA, and vascular remodeling. It leads to the arterial stiffening or arteriosclerosis, renal fibrosis, cerebrovascular disease or stroke, and so on other diseases. Okay. So vasoconstriction is caused by the action of angiotensin 2, not angiotensin 1. That is why number B is incorrect. So is it clear or should I repeat it? Is this problem number 12 clear to all, or should I repeat this thing? Okay, I will repeat it one more time. It may seem to be difficult at first. Okay, so this let us first focus on this renin-angiotensin aldosterone pathway one more time. Okay, so sometime what can happen, like due to dehydration or sodium deficiency or hemorrhage, or some say these are the several reasons. For that, the blood volume may decrease, which can lead to the decrease in the blood pressure. Okay, then decreased blood pressure. So when once the blood pressure is decreased, how our body reacts to make it to the normal? by increasing the blood pressure. So what does happen? Once the blood pressure is decreased, it directly activates the juxtaglommular cells of the kidneys. Now the cells of the kidneys, they produces renin, and so renin is a very important component in case of this our renin angiotensin pathway for the increasing the blood pressure. This renin is increasing of the renin it activates the angiotensinogen in the liver, which increases the amount of the angiotensin 1. All these are the peptides, okay? All these are the different enzymes and all these are the peptides. And this angiotensin 1 is then converted to angiotensin 2 with the help of the angiotensin converting enzyme or ACE, which is present in the lungs, okay? Now, this angiotensin 2, it will lead to several things. One is the vasoconstriction of the arteriole. Vasoconstriction of the arteriole increases the blood pressure till it returns to the normal. Okay, it also will activate the adrenal cortex, which increase the aldosterone. Increasing aldosterone will increase the in kidney. Will increase the sodium and water reabsorption, which will indirectly increase the blood volume and increase the blood pressure till it is going to the normal. Okay, so now the statement, now let us see the statement. Statement number A says that aldosterone is a steroidal hormone. It is correct. Statement number B says vasoconstriction is caused by the action of angiotensin 1. This is false because vasoconstriction is caused by the action of angiotensin 2. Angiotensin 1 cannot do the vasoconstriction. Okay, the aldosterone enhances the retention of sodium and water in blood. It is also correct. And bradykinin is destroyed by ACE. It is also correct answer option. Okay. So now is it correct? How it is now it is clear how the renin angiotensin aldosterone pathway it actually works out. Is it clear or should I discuss it more? Let me know in the chat box if it is clear to you, or should I repeat it one more time? If there is no doubt, okay, okay, good. Now I will then move to the next problem if it is clear to all. Now let us move to the next problem. Problem number 13 says which of the following amino acid pairs is removed from angiotensin 1 by ACE? Your options are number A, histidine and proline, number B, aspartate and arginine, number C, histidine and leucine, and number D, valine and tyrosine. Which of the following amino acid pairs is removed from angiotensin 1 by ACE? I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer for problem number 13. Which of the following amino acid pairs is removed from angiotensin 1 by ACE? Let me check the chat box. I have to be a little quick from now on because we have a couple of questions left. Okay. I have got two answers. One is C number and another is B. Let me check which of the following is the correct answer for problem number 13. So the amino acid pair that is removed by from angiotensin 1 by ACE will be, it is number C, histidine and leucine. So angiotensinogen is converted to angiotensin by the action of renin. Okay, angiotensinogen is converted to angiotensin 1 by the action of renin. Angiotensin 1 is converted to angiotensin 2, which is a potent vasoconstrictor by the ACE or angiotensin converting enzyme. So this is the pathway with the biosynthesis of angiotensin 2. So angiotensin 1, it is basically a decapeptide. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So as you can see, 10 amino acids are present. Angiotensin converting enzyme, it actually clips the last two amino acids, and this is the octapeptide, which is the angiotensin 2, and the amino acid pair which is removed from angiotensin 1 to convert it to angiotensin 2 is the histidine and leucine. So angiotensin converting enzyme is a key component of the biosynthetic pathway that generates a hormone angiotensin 2. The pathway involves the conversion of angiotensinogen to angiotensin 1 catalyzed by the enzyme renin, followed by the conversion of the angiotensin 1 to angiotensin 2 catalyzed by ACE. Angiotensin 2 is potent vasoconstrictor which results in the increased blood pressure. So drug that block the synthesis or actions of this hormone can act as the antihypertensives. At present, there are three main categories of the drugs affecting the biosynthesis or action of the angiotensin 2, renin inhibitor, ACE inhibitor, and angiotensin 2 receptor antagonist. In the so this is a different thing. The design of the ACE inhibitor demonstrates how it is possible to design the drugs for the protein target in the rational manner, even if the structure of the target has not been determined. ACE is a membrane-bound enzyme which has been difficult to isolate and study. It is a member of the group of the enzyme that is known as the zinc metalloproteases that catalyzes the hydrolysis of the dipeptide fragment from the end of the decapeptide called angiotensin 1 to give the octapeptide angiotensin 2. Angiotensin 2 is an important hormone that causes blood vessels to constrict, resulting in a high rise in the blood pressure. Therefore, ACE inhibitors are potential antihypertensive agents because they inhibit the production of the angiotensin 2. Although the enzyme ACE could not be isolated, the design of ACE inhibitor was helped by studying the structure activity mechanism of another zinc metalloprotease, an enzyme called carboxypeptidase. Okay, so this was about the angiotensin 1 and 2. Now let me go to the next problem. Problem number 14 says losartan is a dash type of antihypertensive agent. Your options are number A diuretic, number B beta blocker, number C ACE inhibitor, and number D AT1 blocker. Losartan is a which type of antihypertensive agent? Number A diuretic, number B beta blocker, number C ACE inhibitor, and number D AT1 blocker. I will wait for a minute, then I will check the chat box. Let me know A, B, C or D which of the following is the correct answer. Losartan is which type of antihypertensive agent? Let me check the chat box. Okay, I got two answers. One is saying number D, one is saying number C. Let me check which of the following is the correct answer. So losartan is correct answer is number D, AT1 blocker. Okay, very good. Number D was in the chat box. So antihypertensive agents are of the different types. When we have diuretics like thiazides, hydrochlorothiazide and chlorothiazide, chlorothiazidine, then high ceiling like furosemide, caplacpurin like spironolactone and amiloride. So they act as on kidneys to increase the excretion of sodium and water, decreasing blood volume, decreasing blood pressure. Angiotensin converting enzyme or ACE inhibitors the captopril, linezolpril, enalapril, ramipril, fosifril. Mechanism of action: inhibit the synthesis of angiotensin 2, decrease in the peripheral resistance and the blood volume. Angiotensin 81 blocker are losartan, candesartan, valsartan, telmisartan blocks the binding of angiotensin 2 to its receptors. Adrenergic receptor antagonists or beta blockers like propranolol, nebivolol, atenolol, comparative antagonists that block the receptor sites on the adrenergic beta receptors. So how they work? 81 blockers inhibit the renin-angiotensin aldosterone system, which is also known as the RAS or RAAS, a hormone system that regulates the blood pressure and fluid balance. Block angiotensin 2. Angiotensin 2 is a powerful vasoconstrictor. It narrows the blood vessel, prompts the release of the aldosterone, which causes the body to retain sodium and water. Bind to the 81 receptors. 81 blockers specifically bind to and block to the 81 receptor, preventing the angiotensin 2 from activating them. Resulting effect: the blocking action causes blood vessel to relax and widen, means vasodilation, which lowers the blood pressure and reduces the workload on the heart. Okay. So this was the problem number 14. Now let me go to the next problem. Problem number 15 says the correct statement among the following regarding the antihypertensive drug enalapril is: Number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability. Number D, all of the above. Which of the following statement is correct regarding the antihypertensive drug enalapril? Number A, it is an irreversible inhibitor of angiotensin convertive enzyme. Number B, it is a prodrug. Number C, it has a poor bioavailability. And number D, all of the above. Again, I will wait for a minute, then I will check the chat box. The correct statement among the following regarding antihypertensive drug enalapril is: Number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability. And number D, all of the above. Let me check the chat box now. Okay, I got two answers, number D and number B. Now let me check which of the following is the correct answer, number D or number B. So the correct answer will be, it is number B. It is a prodrug. Very good. So this is enalapril, which is a prodrug. It can cross the membrane, and this is the prodrug because it has, as you can see, there is a ester here, and the active drug is the enalaprillate, which is the acid, which is the antihypertensive agent. So enalapril is a product, and enalaprillate is the active form. So how it works? Enalapril is a drug, mean it is not active until the body converts it. After ingestion, the body converts enalapril to its extreme form enalaprillate. It inhibits ACE. Enalaprillate inhibits the angiotensin converting enzyme. This prevents ACE from converting angiotensin 1 to angiotensin 2, a powerful vasoconstrictor. It relaxes the blood vessel by blocking the formation of the So am I audible? Okay. Hello. Okay, sorry. Hello. Hello. I'm very sorry there was a network disruption. So, am I audible? Please let me know in the chat box if I am audible or not. Okay, okay. Sorry, so there was a network disruption. So that is why I'm sharing the slides one more time. Okay. So is the slide visible? Can you let me know if the slides are visible or not? Okay, sorry, there was some network issues. So I hope now it is fine. Okay. Now let us continue where we were in between. Okay. So I was discussing about the enalapril, right? So basically, enalapril is an antihypertensive agent, but it is a prodrug. The active drug is the enalapril, which is converted. So enalapril is converted to the enalapril after the injection in our body. Okay, and this enalapril inhibits the ACE. This prevents ACE from converting angiotensin 1 into angiotensin 2, a powerful vasoconstrictor. It relaxes the blood vessel by blocking the formation of angiotensin 2. Enalapril helps to relax and widen the blood vessels. This reduces the overall pressure and makes it easier to heart to pump blood. Reduces the stress on the heart. With the less resistance on the blood vessel, the heart does not have any work as heart. It reduces the risk of the heart attack, stroke, and kidney damage. So this is the enalapril, how it binds in the active site of this AC inhibitor. Okay, there are several pockets. One S1 pocket is there where this side chain is goes to. There is a S1 prime pocket. There is a zinc which is present in here with which this carboxylate side binds, and this carboxylate on this proline binds with the arginine of the active site. Okay. So I hope it is clear to all. If it is not clear, I will repeat it one more time because there was some network issue, and I think it is visible to you. If not, please let me know. Okay. Let us go to the next problem then. So problem number 16, according to the fragment-based drug discovery, which of the following parameters are incorrect for the fragments to be used in the lead compounds? Your options are number A, molecular weight greater than 300, number B, no more than 3 rotable bonds, number C, log P value equal to 3, and number D, no more than 3 hydrogen bond donor or hydrogen bond acceptor. According to the fragment-based drug discovery, which of the following parameters are incorrect for the fragments to be used as the lead compound? Let me know A, B, C or D which of the following will be the correct answer for problem number 16. I have to be a little quick from now on. So which of the following will be the correct answer for problem number 16? Let me check the chat box. Okay, I got two answers. One is saying it is number A, other is saying it is number B. Let me check which of the following will be the correct answer, number A or number B. Or is there any different answer? So the correct answer will be, it is number A, molecular weight greater than 300. Okay, so properties of the lead compound, it is very necessary. Some of the lead compounds that have been isolated from the natural sources have sufficient activity to be used directly in a medicine without serious side effects. For example, morphine, quinine or paclitaxel. However, most of the lead compounds have low activity and or unacceptable side effects, which means that a significant amount of the structural modification is required. If the aim of the research is to develop an orally active compound, certain properties of the lead compound should be taken into account. Most orally active drugs obey the rules laid down by the Lipinski's rule of 5 or Weber's parameter. A study of the known orally active drugs and lead compounds from which they are derived demonstrated that the equivalent rules of the lead compound should be most stringent. This is because the structure of the lead compound almost certainly has to be modified and increased in both in the terms of the size and the hydrophobicity. The suggested properties of the lead compound are that it should have a molecular weight of the 100 to 350 AMU and C log value of 1 to 3. The as a measure of how hydrophobic the compound is. So in general, there is an average increase in the molecular weight of 80 AMU and increase of 1 in C log P when going to lead compound to the final drug. Studies also show that the lead compound generally has a fewer aromatic rings and hydrogen bond acceptor compared to the final drug. Such consideration can be taken into account then when deciding whether the lead compound is used to the research project if several structures is available. Another approach is making the decision is to calculate the binding or ligand efficiency of each potential lead compound. This can be done by dividing the free energy of binding to the each molecule by the number of non-hydrogen atoms present in the structure. The better ligand efficiency and the lower molecular weight of the final optimized structure is likely to be. Moreover, if you have a choice of the lead compounds, the most suitable one is necessarily the most potent. For the fragment-based lead discovery, a rule of 3 has been suggested for example, a molecular weight less than 300, not more than 3 hydrogen bond donors, not more than 3 hydrogen bond acceptors, C log P equal to 3, no more than 3 rotable bonds, and polar surface area of 60 Armstrong square. Okay. So this was the problem number 16. Now I will go to the problem number 17. If you have any doubt, please let me know in the chat box. I will check in the and I will discuss it in the last. Okay. The problem number 17 says low concentration of the neurotransmitter dopamine has been implicated in the Parkinson's disease and L-DOPA is the most widely used medicine to slow down this disease. Which of the following statements is incorrect? Your options are number A, dopamine cannot cross blood-brain barrier. Number B, L-DOPA acts as a prodrug. Number C, L-DOPA can cross the blood-brain barrier. And number D, L-DOPA is first decarboxylated by dopamine decarboxylase before entering the brain. Low concentration of the neurotransmitter dopamine has been implicated in Parkinson's disease and L-DOPA is the most widely used medicine to slow down the disease. Which of the following statement is incorrect? Number A, dopamine cannot cross blood-brain barrier. Number B, L-DOPA acts as a prodrug. Number C, L-DOPA can cross blood-brain barrier. Number D, L-DOPA is first decarboxylated by dopamine decarboxylase before entering the brain. Let me know. I will wait for a minute. Okay. A, B, C, or D, which of the following is the correct statement is incorrect regarding the L-DOPA? Let me know A, B, C, or D, which one of the following statement will be incorrect for problem number 17. Let me check the chat box if there is any answer because I have to complete discussing the question. So I have two answers, number C and number D. Let me check which of the following will be the correct answer, number C or number D. So the correct answer is number D. L-DOPA is first decarboxylated by dopamine decarboxylase before entering the brain. This statement is incorrect. Okay. So levodopa is an amino acid. Levodopa is the biosynthetic precursor of dopamine. The mechanism, the brain levodopa is taken up by the dopaminergic terminal of the strontium converted to dopamine by the levoaromatic amino acid decarboxylase. Dopamine is cannot be since it does not enter the brain. Levodopa itself is largely inert. Its effects depend on the increased synthesis of dopamine in the brain and are mostly related to the activation of the D2 receptor. Okay. So this is the our blood-brain barrier. So this dopamine cannot cross this blood-brain barrier. Most of the drugs they cannot cross the blood-brain barrier. The brain is safe in this manner. So levodopa is used. So this levodopa it can cross the blood-brain barrier, and inside the brain it is converted to the dopamine and binds with the D2 receptor of the postsynaptic terminal of the strontium. So how it works? Conversion of dopamine. Levodopa is a precursor of dopamine and can cross the protective blood-brain barrier. Once it crosses the barrier, it is converted into the dopamine to replenish the blood's depleted supply to improve the motor symptoms. Then combination with the carbidopa. Levodopa is often combined with another drug carbidopa. A meditation like cinnamate and Rytary. And the role of the carbidopa. Carbidopa prevents levodopa from broken down in the bloodstream before it can reach the brain. It allows the lower dose of levodopa, which reduces the side effects like nausea and vomiting. Okay. So this was all about the levodopa. I will go to the next problem. Problem number 18 says which of the following methods can be used for the lead design? Your options are number A IR, number B NMR, number C ESIMS, and number D GCMS. Which of the following methods can be used for lead design? IR, NMR, ESIMS, or GCMS? Let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 18? Which of the following you think is the method that can be used for the lead design? IR, NMR, ESIMS, or GCMS? Which of the following methods can be used for lead design? Let me check the chat box. Also, we have to discuss it quickly. Okay, I got one answer which says it is number C. Let me check if number C is the correct answer for this problem or not. So the method which can be used for lead design will be, it is number B, NMR. Okay. So recently NMR spectroscopy has been used to design a lead compound rather than discover one. It is in essence the method sets out small molecules or epitopes which will bind to the specific or different regions of the protein binding site. These molecules will have no activity by themselves as they only bind to one part of binding site. But if larger molecule is designed which links these epitopes together, then a lead compound may be created which is active and binds to the whole binding site. This process is also known as the epitope mapping. So this is a protein. Okay. So the protein has a different binding sites. So let's say for the first one ligand is screened, and this ligand is developed by using the NMR. Now this ligand binds to a particular portion or particular binding site of the protein. Then optimization will make it fit to the particular site. Then the same thing happen for another binding site. So let's say there are two fragments of the proteins which fit to different binding sites which are nearby. Now if this both can be joined together, a new and in a single molecule, so a new lead molecule is synthesized. This is known as the epitope mapping. So the use of NMR spectroscopy in finding lead compounds. NMR spectroscopy was used in design high affinity ligand for the FK506 binding protein, a protein involved in the suppression of the immune response to optimized epitope A and B. This is A number A, this is number B. Both uh, to optimize epitopes were discovered which bound to different region or binding sites. Structure C was then synthesized using these two epitopes linked together by a propyl linkage. This compound has a higher affinity to either of the individual epitopes and represents a lead compound for further development. Okay, so this is how NMR can be used for the lead design. Now, problem number 19, it says that cisapride is a gastric agent and it was withdrawn from the market because your options are number A, it lowers the iron absorption of the body. Number B, it has a poor bioavailability. Number C, it inhibits the HERG potassium ion channels. And number D, it invades the blood-brain barrier. Which may be the which of the following should be the maybe the correct reason for the withdrawn of the cisapride from the market? Your options are number A, it lowers the iron absorption in the body. Number B, it has a poor bioavailability. Number C, it inhibits the HERG potassium ion channels. And number D, it invades the blood-brain barrier. Which of the following will be the correct answer? Cisapride, a gastric agent, was withdrawn from the market because of which of the following is the correct reason for its market's withdrawal? A, B, C, or D? Let me know. Let me check the chat box. I got two answers. One is saying number C, other is saying number A. Let me check which of the following is the correct answer for this problem number 19. So the main reason for the withdrawal of the cisapride from the market will be, it is number C. It inhibits the HERG potassium ion channel. Okay, so this is the structure of the cisapride. In the in vivo and in vitro tests are often carried out in the early stage to find out whether the lead compounds or candidate of the drugs are likely to have certain types of toxicity. One such test is to see whether compounds inhibit HERG potassium ion channels in the heart. HERG stands for a gene that codes for this protein, the so-called human ether agogo related gene, who make so this is a very funny name actually. So several promising drugs that have withdrawn from very late stage of the development because they were found to inhibit the HERG potassium ion channels. Inhibition can result from the disruption in the normal rhythm of the heart, leading to the fibrillation, heart failure, and death. Gastric agents cisapride and the antihistamine terfenadine both are the withdrawn from the market because of the problem. A large variety of the other structure have been found to be unwanted effects, so test to detect the property is best done as early as possible in order to remove the property as part of the drug optimization process. Okay. Now, the last question of today's session, it says that the term used to signify the biological effect of the preparation without the active drug that appears to induce beneficial effects similar to that of the active drug preparation is number A, placebo, number B, agonist, number C, me too drug, and number D, peptidomimetic. The term that is used to signify the biological effect of a preparation without the active drug that appears to induce the beneficial effect similar to that of the active drug preparation is placebo, agonist, me too drug, or peptidomimetic. This is the last question of today's session. Try to answer this question, okay? The term that is used to signify biological effect of a preparation without the active drug that appears to induce biological beneficial effect similar to that of the active drug preparation is known as what? Since we do not have much time, let me check the chat box. Let me see if there is any answer because I have to discuss it. Okay, I got answers. One is B and another is A. Let me check which is the correct answer. So the correct answer is number A, placebo. Placebo is the term of this particular preparation. Okay, so placebo is defined as a substance that has no known specific pharmacological activity against the condition being treated. Placebos can be therapeutically beneficial to some patients when they give rise to the placebo effect. What is the placebo effect? The fake treatment that can make someone better simply because they expect it to help. Okay, so as long as the patient believes that it works, it will work. So this is known as the placebo effect. So agonist and antagonist. Agonists are the drugs that occupy the receptors and activate them. Antagonists are the drug that occupies the receptor but do not activate them. They block the receptor by the activation of the agonist. Me too drug, a drug that's specifically very similar to already known drug with only minor differences. The term Me Too carries a negative connotation. However, Me Too products may create a competition and drive the prices down. Me Too drugs can broadly defined as a chemically related to prototype and other chemical compounds which have an identical mechanism of action. And we have the peptidomimetic. These are the molecules that significantly reduce peptide character that mimic the bioactive conformation of the peptide, thus retain ability to interact with the biological target, cause the same biological effect. Small protein like chain designed to mimic peptide arise from either the modification of the existing peptide or by designing similar type of mimic like peptides or beta peptides. So this was the last question of today's session. So today majorly we discussed about the drug discovery processes and we discussed about the lead molecules and various aspects of the clinical trials and preclinical trials. So I will share my email ID. If you have any doubt or any query, please let me know. via email. So this was our week 9 live session. And today, so whatever, if you have any whatever doubt you have regarding today's session or this class material, please let me know. In next class you also can let me know, or you can let me know via email also, which is which will be better if you let me know via email, so that I can prepare a particular slide for the same. So I will be closing the session if you don't have any doubt. So thank you all for joining. Today it was our week 9 live session. Let us meet on the next Friday for our week 10 live session for this NPTEL course of Organic Chemistry in Biology and Drug Development. Thank you all for joining. I will sharing this class material and the recording after the class. Okay. So thank you. I am closing the session.